Good. Okay, so actually just before, right now it's 4 o'clock, so we should start this preclinical translational session. So first of all, me and my co-chair, Celeste Simon, would like to thank all of you for coming. We would also like to thank the Kidney Cancer Association for sponsoring this 2 preclinical translational sessions this year. This is the first time KCA actually has doing this pre-conference as well as focusing on basic translational research with 2 sessions focused on some of the critical questions in kidney cancer research. So we are very excited. We also want to thank all of the speakers who agreed to speak in this session. So in the interest of time, we would like to keep this session start as soon as possible. So for preclinical session number 1, we have 5 speakers. Each speaker will get 10 minutes presentation plus 2 minutes question and answers. And then towards the end, we're going to have about 10 minutes Q&A sessions that could help people answer some of those overall questions for all of the speakers. So without further ado, I would like to introduce the first speaker, David Brown from Yale University. Thank you.
Thank you so much for the invitation to speak, and it's good to see everyone today, and happy to kick off this meeting. So I'm going to speak today about understanding and optimizing anti-tumor immunity, but with some new data, particularly about steering the immune response. And so the learning objectives we'll get to is really thinking about antigen-directed therapies, a steering wheel sort of in anti-tumor immunity in kidney cancer. And so I've used this slide before to sort of introduce the idea of what are the tools that we have to induce anti-tumor immunity in kidney cancer. And we think of it as essentially a car of T-cell immunity and trying to get that as quickly as possible towards the tumor. This is really a nice simplified framework that Kathy Wu and Patrick Ott have used in the past. And when we use this, we can think of our current tools and how they sort of play into this framework. So we can release the brakes on this car and let it go towards the tumor, and that's immune checkpoint inhibitors. We can press on the gas pedal, and that's classic cytokine therapies and new engineered cytokines. None of that gives directionality, though, to the immune response. And so can we actually steer the immune response towards the tumor specifically by adding antigen-directed therapies? And of course, there's other ways of modulating T-cell intrinsic factors. And so what I really want to talk today about is the sort of adding a steering wheel component. And there's lots of ways to direct immune responses towards tumors. And I tend to think of it along these 2 axes. Are you targeting something on the surface or targeting something that's intracellular, that's sort of then chopped up and presented in an HLA-restricted fashion? And is this something that's personalized or something available off the shelf? And so as we think of these 2 axes, we can fill in the different buckets. Something like conventional CAR T cells, they're personalized, they target surface antigens. Other things like allogeneic CAR T cells are, again, targeting surface antigens, but are off the shelf. There's HLA-restricted targeting. And what I really want to talk about is this last bucket, which is how to target HLA-restricted epitopes really in a personalized way. And why would we want to do this? Why would we want to target things like neoantigens in kidney cancer? And I think there's a set of rationales for this that at least support the hypothesis they might be a reasonable target. We know that in general, neoantigens can be effective targets of anti-tumor immunity across a variety of cancer types. There's now data in melanoma and glioblastoma and pancreatic cancer that these neoantigens are targetable. We know kidney cancer is historically and contemporarily immunogenic, and that while adjuvant therapy exists right now, there's certainly a need to improve these therapies. And so we asked the question, can we really design an immunologically effective personalized neoantigen vaccine in kidney cancer? And so to do this, this was an initial pilot study of 9 patients. We selected patients with stage 3 or stage 4 kidney cancer that had fully resected, so they had no evidence of disease. We predicted based on sequencing what would be the most likely to be immunogenic epitopes. We synthesized up to 20 of them using synthetic long peptides, and then we put them into 4 pools, so up to 5 antigens per pool, and then vaccinated in a prime boost format. So the first question really was just, can we actually do this effectively? Kidney cancer has a very modest mutation burden. It's not melanoma. It doesn't have hundreds or thousands of mutations. So can we actually make a vaccine for each patient? And the answer to that was yes, that despite having a modest mutation burden for every single patient, we're able to successfully manufacture a vaccine. And we're targeting both single nucleotide variants and also frame shift insertion deletions. And for many patients, actually able to target RCC driver mutations. The next question was this able to effectively elicit an immune response? And so this is a lot of data that I'm going to try to break down, but the way we assessed this was by interferon gamma ELISpot. So we took patient blood samples prior to vaccination, during vaccination, and after vaccination, and then ex vivo added peptides and saw whether that elicited a T cell response by the secretion of interferon gamma. And what we can see in this heat map is a lot of data that's summarized. But if we go across the first column, that's 0, that's the pre-vaccination time point, and it's essentially all white. There's no pre-existing immunity to the neoantigens we vaccinated against. But as we move from left to right across that heat map, we see all of a sudden that in every single patient, at least for one of the vaccine pools, we were able to elicit anti-tumor or elicit an immune response by interferon gamma. So if we look at one example patient, the first patient enrolled in the study, for each of the vaccine pools, we can track over time, moving from left to right, the induction of vaccine-specific immunity. And in this case, 3 of the 4 pools elicited strong immune responses. We can then go further, and for each of those individual vaccine pools, we can arrest the T cells and then re-stimulate them with one of the peptides to see which of the specific peptides containing neoantigen are immune responsive. And we can see that even within a peptide, even within a vaccine pool, there's tremendous diversity. There's some pools which aren't responsive at all. And we see, for instance, pool B in this case, not eliciting a strong response. But then even within a pool that elicits a strong response, it's highly variable. So in pool D, a mutant VHL neoantigen elicited a very strong immune response, whereas not all the peptides in that pool did. So I think our first question was, is this effective in generating an immune response? And in all 9 patients, yes, it was able to generate an immune response. The next question was, which are the targets that led to the best immune response? And it really seemed to be the clonal driver mutations, for the most part, that elicited strong immune response. I'm showing an example interferon gamma ELISpot, showing the response of VHL mutant peptide compared to 2 negative controls and a positive control, PHA, and we can see incredibly high reactivity against mutant VHL. If we look at common RCC driver mutations, with one exception, the far majority of them were able to elicit immune responses. And in general, if we look at pan-cancer drivers, the majority of driver mutations were able to elicit immune responses, whereas only a minority of passenger mutations were able to do so. The next question was, are these, we know we can get immune responses, are these durable immune responses, and ultimately do they lead to anti-tumor reactivity? And so this was a bit of a complicated workflow, but I'll try to sort of walk through it. The way we assessed this was twofold. One is we performed delayed type hypersensitivity assessments in all of these patients. So this is the equivalent of a PPD test that you might get for tuberculosis, where we take all 20 peptides at small concentrations without any adjuvant, and inject them intradermally under the skin of the forearm, and then wait 48 to 72 hours. And if there's an immune response, it's because there's an influx of T cells. And that's what we saw. This is an example patient, but really we saw this response in all patients. We could then biopsy that skin site and perform single-cell RNA and TCR sequencing to see actually what are the TCRs, T cell clonotypes that are flooding in that are presumably vaccine reactive, and that's what's shown in this area here. So this has enabled us to think about what are vaccine-reactive T cell clonotypes. Then we can actually go to the tumor material itself and perform single-cell RNA and TCR sequencing. And using signatures that have been previously generated in Kathy Wu's lab, we can roughly characterize T cells as either being tumor-specific, using, again, a particular signature, or non-tumor specific. And now we essentially have barcodes. We have barcodes for T cell clonotypes that are tumor-specific, virus-specific, and then also vaccine-specific. And we can use that to really track those T cell clonotypes throughout the blood. And when we do that and we track over time, we can see that the viral-specific clonotypes really are fairly stable. The ones that we suspect are tumor-specific clonotypes in purple, again, fairly stable. But when we look at the vaccine-specific clonotypes in the peripheral blood, we see this really high upregulation, over 100-fold upregulation with vaccination. And while it dips, that increase actually persists for a really long amount of time. That actually, we stopped the last vaccine is at 20 weeks. And if we go out to really 2 to 3 years later, we still see many of those clonotypes persisting. Again, arguing that this is inducing durable T-cell immunity. The next important question is, okay, it's getting an immune response, it seems to be fairly durable. Is this actually hitting tumor, or is this just a random immune response against peptides? And so what we could do is take those T cells, those post-vaccine T cells, and then culture them, expand them with vaccine-specific peptides, and then add them back on top of the patient-matched tumor, the autologous tumor, to see if there's reactivity by interferon gamma ELISpot. And that's exactly what we see, that for not every patient, but for 7 of 9 patients, there was anti-tumor reactivity in these neoantigen reactive T cells, arguing again that these are actually capable of tumor recognition. And then the final question is, was there any at least signal of clinical efficacy? And I'm almost hesitant to show it because, again, this is 9 patients. It's a high-risk cohort, stage 3 and 2 stage 4 patients, but it's 9 patients that could be statistical flukes, all of these sorts of things. But at least in this initial cohorts, the results look fairly nice, that none of these 9 patients had a recurrence. All 9 patients were disease-free at the time of cutoff. 1 patient unfortunately died during COVID of an unrelated cause, but at least in these 9 patients, at least a promising sort of early signal that there might be some clinical activity, but again, very small numbers. And so to summarize, I think this argues, one, that neoantigens really can be effective targets of anti-tumor immunity in kidney cancer. And I think this really sets up now a further exploration of this in terms of efficacy in kidney cancer. And that's exactly what's happening with the phase 2 trial, the V940-004 INTerpath 004 study. And so many people to thank, huge collaborative effort from really across Boston and beyond, particularly Tony Schwery, Kathy Wu, Patrick Ott, Darren Keskin, and many others from Broad, MIT, and other places. Our funding sources, particularly Gateway and the CDMRP, and then of course these patients with a huge amount in this phase 1 study, and so really grateful to them and their families. And I'm happy to take any questions. Thank you very much. Good pick. Oh, yeah. Yeah. Yeah, it's a good question. We try to assess the patients, were there determinants such as HLA zygosity or things like that, number of vaccine peptides. There wasn't anything that was sort of obvious that would say this would correlate with magnitude of response or not. It's a good question. I guess not necessarily, and this is based on some of the work that's done out of melanoma, which compared essentially the levels and immunogenicity of tumor-associated antigens expressed at high levels and neoantigens expressed at much lower levels. And I think the sort of end result was that TAAs are expressed at much higher levels, but generally have essentially listed low functionally avid T cells, whereas neoantigens typically listed very functionally avid T cell responses. So even at low levels, they're able to sort of generate responses. And so I think keeping that in mind, maybe not entirely surprising that it's able to generate these responses. Yeah. No, both great point and great question. So Samir Khalif had led some of those early VHL studies, which were really impressive. I think, one of the advantages of having a multi-epitope vaccine is sort of maybe this ability to avoid an immune escape by sort of downregulation of a particular epitope. The question of whether there's pre-existing responses is a really tough one because it's also a limit of sensitivity, limit of detection issue or sensitivity issue. Are there responses there, but we just can't detect in the peripheral blood? Maybe there are pre-existing responses in the draining lymph node or other sites that we're not catching, even with a highly sensitive assay like ELISpot. But I think it's not entirely surprising. I think when we think about how vaccination works, one possibility is basically expanding the repertoire that's already there, just building up the numbers. But the other is sort of new priming that's essentially diversifying the repertoire. And my guess is it's some combination of both. So the fact that we're not seeing that as pre-existing sort of immune responses perhaps is not entirely surprising. But you're absolutely right. It's possible in an immune checkpoint inhibitor treated context, maybe some of those would then become detectable.
Okay, thank you very much. Unfortunately, I think we're going to have to go on, but I'm sure David would be delighted to answer more questions
Thank you so much.
after the session and during the rest of the meeting. So I'll introduce the next speaker, Abhishek Chakrabarty, who is coming to us from Case Western Reserve and actually drove here. It took 6 hours, right? So
because of exit error.
missed an exit. Welcome.
All right, well, that's science, right? You always take the detour. So my lab, we study a lot of, we do a lot of work in identifying new preclinical targets in RCC. And what I'm going to talk to you all today about is a particular amino acid transporter called SLC1A1. It's also known as EAAT3. Neurobiologists have studied this for a long time, but you'll see why this became relevant to us in the kidney cancer space. So what are we going to try to learn today is how epigenetic dysfunction can mark novel oncogenic targets in kidney cancer, particularly solute carriers like SLC1A1, which support metabolic oncogenic programs. We are also going to show you some structure function studies that have allowed us to couple medicinal chemistry with classical biochemistry and identify really nice translational discovery pipelines. The actual paper is currently under review. I also have a preprint in the bioarchives. Everybody can access this. So I'm not going to go into a lot of details into the background because I want to share with you all some of the new unpublished stuff today. But the origin of the story really was when we were looking at epigenetic dysfunction in ccRCC, we had identified this particular epigenetic mark called the H3K27 acetylation mark as particularly elevated in VHL deficient HIF-high ccRCCs. And this mark typically is associated with genes that are switched on across the genome. And so a simple question to ask was, what was H3K27 acetylation driving up in the tumorigenic VHL deficient cells? Were they critical oncogenic drivers that we could identify? By looking at the genes that were highly marked with H3K27 acetylation under tumorigenic versus non-tumorigenic conditions, and then coupling that with a functional readout, we were able to fish out this SLC1A1 transporter, and that's all been described in this original paper. So I'll just show you some of the snippets of the data. So if you actually look at cell lines, we can make cell lines isogenic where they either have VHL or don't have VHL. The blue spectra are H3K27 acetylation peaks in the VHL deficient cells. The gray ones are in the VHL proficient cells. As you can see, the gray bars are much, much smaller. The VHL proficient cells have lower K27 acetylation levels at the SLC1A1 locus. What's also interesting is these same regions are also depressed in normal kidney compared to the patient tumors, which have higher K27 acetylation at these loci. We can also see at an expression level, there is an effect of VHL introduction into these cells. But what's really cool about this is we can add back pharmacological agents that bring up the HIF target genes, such as the hypoxia mimetics. And when you do that, you can get a restoration of HIF and HIF target genes like NDRG1, but SLC1A1 is not restored. In other words, VHL is regulating SLC1A1, but in a HIF-independent manner. So this becomes really relevant from the clinical context. This could still be a viable target in tumors that are HIF2 independent. So we have also done additional experiments where we have done sufficiency. We have added either HIF1 or HIF2 DPA mutants, which are chronically stable, back into VHL proficient cells. We have also done the necessity experiments where we have inactivated ARNT, which is HIF's obligate binding partner in VHL deficient cells. In all conditions, we can convincingly demonstrate that SLC1A1 is not being regulated in a HIF-dependent manner. What was also interesting was when Sabina Signoretti was helping us out with analyzing where SLC1A1 expression goes up in kidney cancer cells, she found that in CCRCCs, the metastatic lines were metastatic tumors were especially having higher levels of SLC1A1. So from a mouse model context, we used the 786M1 cells. These are classic models of metastatic proficient tumor lines that are developed in Joan Massacre's lab. And here again, we can see that you inactivate SLC1A1, and there is a measurable decrease in metastatic seeding and outgrowth. So all of this really bodes well for going in the direction of targeting SLC1A1. We then profiled what normal tissues look like. So these are just mouse tissues. The predominant expression of SLC1A1 was detected in the brain and in the kidney, which meant there was tissue restricted, probably would not have systemic effects if we inactivated it. The neurobiologists have been studying this for a long time. And one of the stories that had been published recently had identified this molecule, which was called compound 3E in the paper. And they proposed that this molecule was actually acting on target and specifically on SLC1A1 and not on the other SLC1A family members, which are very similar to SLC1A1 actually. The problem was that we are really very skeptical in my lab, and essentially there's a logic test, right? So walks like a duck, talks like a duck, quacks like a duck, must be a duck. This molecule fails the duck test. It does not look like aspartate, does not look like glutamate. There is no reason structurally that this molecule should be targeting SLC1A1. And this really confused us a lot. Why the hell is this molecule called a selective molecule? And so we did a lot of rigorous testing to validate that this molecule was working on SLC1A1 and therefore could be developed further. So one of the first experiments that we did is this is a really cute experiment that you can do. You can take cell extracts or even recombinant protein. You can expose them to your drug of interest. If your molecule binds to the protein becomes more heat stable, more thermostable. So this is called a CETSA assay or a cellular thermal stability assay. And here you can see on the top are cells that were treated with DMSO or cells that were treated with this compound 3E. And when you treat with this compound 3E, SLC1A1 is stabilized, suggesting that the molecule is directly engaging with SLC1A1. We then also worked with Olga Butker's lab. Olga Butker has studied glutamate transporters for many years. She's at Cornell Medical College. And with them, we used recombinant human protein. And we did a recombinant version of this assay. And here again, we're looking for an increase in temperature, showing a stability increase. And we found that indeed happens when you expose SLC1A1 with this molecule, which is now called CCA59004 in our pipeline. But what was also equally interesting is that this only happens if you do the experiment in the absence of sodium. This entire transport reaction is sodium coupled. So what we think is happening is this molecule kicks off the sodium from a hidden binding pocket, and that's how it's blocking the activity of this transporter. So is this working by mechanism that you would expect it to be true? So we treat cells with this molecule, we do metabolomics analysis, and now you start looking for what are the changes that are occurring at the steady state level. As you would expect, aspartate and glutamate, which were the canonical substrate of SLC1A1, those molecules as well as the metabolites that are derived from those are the ones that are depleted in cells when you inactivate SLC1A1. But what's also equally interesting is that you take these cells. Normally you treat these cells, in the top are the untreated cells. You treat the cells with the compound and the cells start to have a fitness defect. But you now provide exogenous sources of either esterified aspartate or glutamate, or you provide exogenous glutathione, which is coming from the glutamate and cysteine backbone, and now you can actually reverse the cytotoxic effect, at least partially, suggesting that the effects of this molecule, the cytotoxic effects are at least working in a metabolically on-target manner. So what do we do about this? How do we develop this further? We have now solved the cryo-EM structure of this molecule in complex with SLC1A1. We have a very high resolution structure. We know exactly where it sits. It's not in the aspartate glutamate pocket, as you would imagine. It's actually in the sodium binding site. There's a cryptic site that gets exposed in the presence of the molecule. We can now make mutations in the residues that are engaging with this particular inhibitor. And we can ask the question, are these going to be sufficient to drive resistance to the drug? One way we first test that the mutation itself is not going to be deleterious to the protein is we can take cell lines, we can inactivate SLC1A1. For example, in the first column, I keep doing the same thing. In the first column, you can see we inactivate SLC1A1, the cells start to die. You add back wild-type protein, the cells can live in the presence of the CRISPR guide. But now you can use this assay to score whether a given mutant is active or inactive. So for example, from this assay, you would say that F99A, this mutant is not functional, but the other mutants are all functional. They can be tested in your drug resistance screen. And when we test these, we can actually find that there are critical sites that are engaging with the molecule, which when you mutate them, now start conferring resistance to the molecule. And one example of that is this F99L and F99M mutant. You put these mutants into the cell, you expose the cells to the drug, and the drug does not have as much of an effect as it has on the GFP or the wild-type expressing control cells. So all of this means that it's all working on target, selective molecule for SLC1A1. The medicinal chemists are really excited about this idea, and they have really gone and literally hit every single part of this molecule that they think is a weakness. They've come up with a list of about 50 molecules right now. They're making 50 more. We have started drug screening with a number of these molecules. At least 8 of them right now in initial assays are showing greater potency than the original molecule that we started working with. That's the benchmark that we are working against. Out of the 8, I think 3 of them, we have some validation that they're also working on target, probably improving potency and also having better pharmacological characteristics. But we also want to diversify our molecular portfolio a little bit. We don't want to bet all our money on this particular backbone. And what we have done is we've been a little ambitious with this. And we have started doing what's called these DNA-encoded libraries. The idea is that these are small molecules with a DNA tail on them. The barcode on the DNA allows you to sequence the molecular identity itself in downstream assays. So we are using recombinant SLC1A1. We are exposing it to this library of about 3 billion molecules now. And we are using the DNA as a barcode to fish out the molecules that are able to directly engage with SLC1A1 with the hope that at least a few of them will be potent inhibitors of SLC1A1 down the line. So this entire pipeline is now ongoing. All right, so what does this mean for us? So question is, what does it mean for my practice on Monday? It depends on which Monday you're talking about, because preclinical work takes, it's a marathon. It takes a long time. And so perhaps nothing for the coming Monday, but stay tuned. We keep pushing hard on discovering new preclinical targets to work on, and finding potentially new translational vulnerabilities to block. So SLC1A1's metabolic function is a HIF-independent translational vulnerability that we are now going after. I also want you all to take home the message that this entire pipeline, where you can combine structure and biochemistry and cell-based studies together, really creates a really nice synergistic environment for making some really cool drug discovery pipelines, okay? I really want to make a pitch for how excited I am over the fact that there was a preclinical session this year. And really, I cannot thank you all enough for being here in these numbers and actually giving us the platform to come and talk to you all about this. I'm really, really sincerely grateful for this. So I'll take any questions.
All right, one burning question. So just very quick, did you test those compounds in vivo?
So in vivo, so I didn't talk about it much, but there's furan on that molecule that's really bad for in vivo use. So the idea is that as soon as we get a molecule without that furan ring, we go in vivo. The CCF59004 was tested in vivo. The problem was it accumulated in the brain, which we do not want to happen. We want it to go preferentially to the kidney, and so hopefully new molecules will fix that.
Okay, great. Thank you, Abhishek.
Okay.
So, all right, next speaker is actually our keynote speaker tomorrow, Celeste Simon from UPenn. She's graciously enough to actually give 2 talks today, but along the same topic of targeted metabolism in kidney cancer. Thank you.
Thank you. And this is along the same theme. And our aim, potentially overly ambitious and somewhat naive, is to see if we can identify what we believe are essentially universal metabolic targets that we can target and drug in renal cell carcinoma. And this is an admittedly genetically diverse disease. It has profound, not only intertumoral heterogeneity, but intratumoral heterogeneity at every level I can think of. But the idea is, can we find properties of this disease that we detect in every single patient that we might be able to exploit? And so that has been our approach for about a decade now. And the obvious place to look, amongst other pathways, is to interrogate something that I think everybody in the room appreciates, is that this disease accumulates astonishing quantities of neutral lipids, such as triglycerides, cholesterol, cholesterol esters. In fact, this makes them strictly dependent on lipid droplet formation. And this is why we call the tumors clear cell renal cell carcinoma upon histological investigation. And so with this in mind, we have found what we really think are what we've been looking for. Proteins that are detected in every single tumor sample we have interrogated. And one is the HDL cholesterol lipoprotein transporter called SCARB1, which I will talk about tomorrow. And what's interesting about SCARB1 is that we believe actually, we believe that this actually contributes to the cholesterol-laden nature of this disease. And a corollary of that is because the tumor cells have so many cholesterol molecules in their derivatives, if, for example, they experience redox imbalance, this will develop what are known as oxysterols, such as 7-alpha-hydroxycholesterol, which can actually be toxic to the cells and lead to cell death. And upon investigating this, we have identified another protein. It has a long, terrible enzymatic name. It's a sterol dehydrogenase, so I'll just call it, HSD3B7. And the reason why the tumor cells are so dependent on this enzyme is that this is how they deal with the cholesterol-laden nature of the cells, because they're prone to developing these lipotoxic oxysterols. And so if you don't think about HSD3B7 every day, it's actually part of the bile acid system. Obviously, bile acids are how we absorb lipids from the diet, and bile acid synthetic machinery can be triggered to limit free cholesterol, in particular oxysterol toxicity. And so we interrogated our own patient samples and many other databases, and we actually noted that the bile acid synthetic machinery, in particular HSD3B7, is consistently overexpressed in hundreds of tumors that we have interrogated. And again, this is the enzymatic reaction that it metabolizes to avoid a buildup of toxic sterols such as 7-alpha-hydroxycholesterol. We did integrative analysis using a number of transcriptomic data sets, proteogenomics, and metabolomics, and we were able to show that, as I mentioned, that it's overexpressed at the RNA and protein level in every single tumor type we've interrogated, even at early stages. And even though it's overexpressed in essentially all tumors we've looked at, there is some intertumoral heterogeneity, and the tumors that tend to have higher levels of HSD3B7 are found in patients with worse clinical outcomes. This also coincides with the fact that the tumors pretty consistently overexpress this product of HSD3B7. It has a long name, so I'll just call it 7-HOCA. And again, this is a way to detoxify these potentially death-promoting oxysterol compounds in these tumors. So another thing that's interesting to note is in a patient cohort of approximately 49 individuals, urinary analysis indicated that these patients have a buildup of bile acids like cholic acid, almost reminiscent as if they've have overt liver injury. And so this would suggest that the pathway and its products could even serve as some kind of biomarker for the disease, something that we're thinking about. So HSD3B7 is very consistently overexpressed in the tumors. Does it mean anything functionally? We did obvious genetic approaches with CRISPR or short hairpins, and in this case, this is a simple flank tumor model. We're letting the tumors get to a certain size and then inducing the hairpin to deplete HSD3B7 and actually causes the tumors to essentially stop growing. Now, is this because of the accumulation of the oxysterols that I mentioned, like 7-alpha-hydroxycholesterol? And some of its outputs can be DNA damage, cell death, and reduced cell proliferation. And so lipidomics approaches indicated indeed that when we eliminate HSD3B7 activity, there is an accumulation of 7-alpha hydroxycholesterol and related compounds, and this is at the expense of producing the 7-HOCA derivative. So we believe that this is actually causing the cell death in the treated cells. We can mimic the buildup of the oxysterols by simply treating the cells with different versions of hydroxycholesterol, where hydroxylations are occurring at different sites, such as 7, 25, and 27. And low levels of these hydroxycholesterols are enough to cause cell death, as shown here by an exon-5 staining or simply looking at the cells. And we can overcome the cell death if we simply overexpress the enzyme. And I should note that we have to overexpress an enzyme that has catalytic activity. So the take-home message here is that we believe because the cells have so much cholesterol, and if they're experiencing things like redox imbalance or other inputs, other stimuli, you can develop these lipotoxic compounds, and they become strictly dependent on this enzyme to avoid cell death. So as Abhishek mentioned, can we exploit this observation? And so we conducted an inhibitor screen. We began with a fluorescence assay, and what we're taking advantage of is that the enzymatic reaction converts NAD plus to NADH. And in a biochemical reaction, we get fluorescence only if we have the purified protein and the precursor 7-alpha hydroxycholesterol and the cofactor NAD+. And then we generated a connected luminescence assay that allowed us to carry out a drug screen. We began this with a library of approximately 1,000 natural compounds as a proof of principle, and we actually identified a number of hits. Actually, one, I rather like the name, cholesterol. And if you look at it, it looks somewhat like cholesterol, so it makes sense that it would actually score in the drug screen. And we found other compounds as well, but cholesterol was the one that was the most cell permeable, so we carried on with that. So once again, if we treat the cells with cholesterol, this absolutely mimics loss of HSD3B7, and we can observe this in multiple renal cancer cell lines, as shown here. Of note, the normal renal epithelial cell counterparts, and we looked at several different types of this, are essentially resistant, but the cells that are dependent on HSD3B7, specifically the renal cancer cells, undergo cell death when treated with relatively low levels of cholesterol. And then finally, we can show that the cells accumulate the precursor 7-alpha hydroxycholesterol, as we might imagine. And then again, in an in vivo system, a simple flank tumor, we get the tumors to a certain size, we treat them with cholesterol, and they stop growing, mimicking genetic loss of HSD3B7. So to step back, I should point out that cholesterol is actually a product of a Chinese herb. It's used to treat obesity. It has a number of other targets, so we're not arguing that this is something that would necessarily be effective in the clinic, although it has undergone some clinical trials, but we are now engaging in a broader screen of roughly 2,000 compounds. We have some hits that we'd like to further characterize. And so the take-home message for everybody is that by the systematic analysis of the unique metabolism of this tumor, we actually have found a couple of potential targets that we have found in every single tumor sample we've interrogated. HSD3B7 is this bile acid synthetic enzyme that the cells are completely dependent on. If they lose it, it results in reduced proliferation and cell death because of a buildup of this lipotoxic compound, oxysterols, specifically 7-alpha hydroxycholesterol, and this underscores the fact that cholesterol homeostasis is essential for renal cancer progression. And so the take-home messages is that, number one, we think this is an example of a universal target that we can exploit. As Abhishek mentioned, this is also a protein that's completely independent of HIF-2 alpha regulation. So again, this could be something that could be combined with HIF-2 alpha inhibitors or used in the acquired resistance to HIF-2 alpha inhibitors. And we would like to suggest that bile acids in the urine could also be in some way a biomarker to monitor early renal cancer lesions. And so with that, thank you for your interest and attention, and I can answer maybe one or two questions if you have them.
For Celeste? Okay, yeah.
I'm just trying to understand, why did the cells do this to begin with? Why do they upregulate SCARB1, end up with all this cholesterol that then is toxic, so then they have to upregulate the
So we think we finally have an answer to that question. So the SCARB1 protein is very consistently overexpressed. In fact, Mendelian randomization studies have indicated that individuals with higher levels of circulating HDL cholesterol are more likely to develop renal cancer. This is because of the SCARB1 protein being expressed. It opens up a number of vulnerabilities because the cells downregulate the endogenous cholesterol biosynthetic system. They become dependent on lipid droplets. They become dependent on the enzyme that I just mentioned. And we believe, and I think there's evidence from other laboratories, is that the cells are prone, because of their lipid-laden nature, to developing ferroptosis, and SCARB1 helps them avoid that. And we think this is going to be particularly important in the metastatic setting.
We think of HSD3B7 as a regulator of cellular homeostasis. What's its physiological, or just thinking about drug development in the future,
Sure.
Where do you see toxicity?
Right, so you're raising a very important question that I thought Jim was going to ask, maybe. So this can be an inborn error of metabolism. Individuals are born with deficits in this enzyme, and they are maintained by being treated with cholic acid and similar bile acid types. And in fact, mouse models of HSD3B7 that have been developed that are perinatal lethal can be kept alive indefinitely by supplementing diet with things like cholic acid. So the toxicity that you might imagine could be a concern. I think there could be ways to manage if it's a problem. So, okay.
That's fine. So I don't need introduction. I can introduce myself. I'm Qin Zhang from UT Southwestern. So today I'm going to talk about some of the ongoing research which I've never presented before on targeting bezutifan resistance in kidney cancer. So this is reflecting our efforts on understanding on molecular level on how we can actually address those either adaptive or intrinsic resistance. So we want to understand how the potential mechanism and then potentially explore some of the therapeutic targets that can reverse this resistance. So that we know there's two kinds of resistance. One is intrinsic resistance. This two paper published back with James Burg-Wallace and Bill Caden's lab published in Nature a few years back, showing some of the CCRC cell lines do display intrinsic resistance because upon bezutifan or PD-2399 treatments, they don't show any cell growth defects. And then also showing the mouse model as well. And then in James Burg-Wallace's PDX model, as here showing on the right, there's actually a lot of, so there's actually plenty of the PDX model which upon bezutifan treatments, they don't actually show the tumor regression. This is intrinsic resistance perspective. Also, there's acquired resistance. For this perspective, obviously, previous research showed the p53 mutants may actually one of the contributing factor for the therapeutic resistance with bezutifan. And then if you, actually this paper by James Burg-Wallace showing that the XP model, the PDX model, upon the long latency of the tumor shrinkage, eventually those tumors start taking off, and then you can see this at the end of the treatment, they start develop this resistance. Obviously, actually the research found found that some of these small population of patients develop this gateway mutation, which kick out those drugs from the HIF-2 HIF-1 alpha binding pockets, which also reflected on the molecular level, showing that some of those extensive treatments induce this actually restoration of HIF-2 alpha HIF-1 beta binding, even in the presence of PD-2399 treatment. So obviously, those are clinical challenges. So how do we address this question? So first of all, I want to talk about adaptive resistance model we have been generating in the lab. So what we did here in this case, we use original, which is responsive cell line 760. So in this case, we actually using CRISPR knockouts, 2 different sgRNA against HIF-2 alpha. In this case, in order to make sure that HIF-2 alpha pathway was completely shut down, so on top of the CRISPR knockout, we also actually chronically treat those cells with PD-2399 at a low dosage. So over 3 to 6 months of time, those resistant clones will develop. And then we can actually pick up single colonies from, from these 96-well plates. And then we can actually look at some of the individual clones. In this case, some of them will be still responsive. In other cases, if you draw the line around here, so some of the resistant clones would regain the growth over time. So those are resistant clones in a long story that we're focused on. So what does that mean, those resistant clones? We actually did perform whole exome sequencing. We did not see a robust mutation in some of the target genes or other pathways. So we think that it could be epigenetic or gene expression change that we might be actually confirm this resistance. So in this case, what we did is actually supported by ACR kidney cancer grants. So we actually performed some of the epigenetic as well as the RNA-seq analysis. In this case, this is showing a heat map that we performed ATAC-seq, H3K27 acetylation, H3K4 monomethylation, as well as RNA-seq. We're comparing those various resistant clones with parental clones. We want to look at some of this gene expression change on the top where the activate gene expression as well as increased RNA expression. Those could reflect some of those oncogenic pathways that eventually confirm the adaptive resistance. So long story short, what we found is actually a short list of genes showing here on the right that could potentially contribute to this resistance. Obviously, we want to see whether, functionally speaking, those genes could mediate the phenotypes. So in this case, we actually overexpress individual target genes and then treat those cells in presence of PD-2399 bezutifan. In the control cells, those cells still responsive toward bezutifan, but when you actually overexpress some of target genes such as PDGS, you actually confirm this therapeutic responsive toward PD-2399, also showing on the right with soft agar growth assay. So this is all pure in vitro. What is in vivo phenotype looks like? First of all, we should understand what PDGS do. PDGS is actually sensitive for BCE2, which mediate inflammation pathways, as shown here on the left. There's also plenty of actually inhibitors that could target this PDGS. So that's something we're particularly interested in. So in this case, we do want to perform some functional assays. So we actually overexpress PDGS plasmid in the 76O cells. In this case, we actually can look at either control cells or another unrelated genes or PDGS. So you can see the control cells actually respond to bezutifan very well, the tumor shrinkage. Another gene, IGFBP3, also showing the tumor shrinkage. But this PDGS overexpression confirms this resistance, at least in the partial manner. So if you look at this tumor growth per se, so those parental cells grow very responsive toward PD-2977 treatments. So it's IGFBP3. But this PDGS overexpression, you can see that actually confirms the resistance to this bezutifan treatment, suggesting at least partially, this PDGS could be actually confirm the partial resistant toward bezutifan treatment, at least in an adaptive fashion. Okay, so this is adaptive fashion we're talking about. The other one will be we look at whether from intrinsic perspective, some of cell lines, especially 50% RCC lines, will have intrinsic resistance toward HIF-2 alpha inhibitor bezutifan treatments. In this case, we would like to actually employ our CRISPR-based approach, where we actually took this UMRC2, which is intrinsic resistance. We infect with the human canon CRISPR library, where we infect the virus and then inject those infected cells in vivo into the mice. We actually treat them with bezutifan or vehicle control. Eventually, we obtain those tumors in vivo and perform the deep sequencing. We want specifically to focus on some of the kinases. Upon its depletion in combination with belzutifan will actually cause tumor shrinkage. That's something we're interested in. We think that some of those kinases will be acting upon in combination with belzutifan leads to tumor shrinkage. So what do we say from this screening? Long story short, we actually found one of those INSR, which is insulin receptor-like kinases. In this case, I just want to show you one of the CRISPR sgRNA. In this case, UMRC2 is resistance. When you inject those cells into the vivo vehicle control, grow tumor readily. If you treat it with belzutifan, as we expect, they don't respond. And then if you actually deplete INSR with CRISPR sgRNA, basically the tumor grow actually just like normal. On the other hand, if you combine the INSR in combination with belzutifan, you can see this tumor shrinkage dramatically. So not only with this sgRNA, we're also showing the similar phenotype with another sgRNA with the same phenotype, suggesting this INSR depletion along with belzutifan and can actually at least partially reverse this belzutifan intrinsic resistance. This is still ongoing. So in summary, I present very two short stories in terms of how to target either intrinsic or adaptive resistance. From adaptive resistance perspective, we did generate the cell line model that can recapitulate HIF2 inhibitor adaptive resistance by performing integrated analysis with ATAC-seq, RNA-seq, ChIP-seq, and RNA-seq. We can potentially identify some potential candidate genes that contribute to this adaptive resistance. And then we're also showing PTGS at least partially responsive towards this belzutifan resistance. And also from an intrinsic perspective, our CRISPR screening approach indeed can identify some potential therapeutic target in combination with belzutifan can reverse this intrinsic resistance. So with that, I want to thank a couple of people in the lab who contribute directly to this project, Fang Zhou and Chen, and also my collaborator from UT Southwestern, including Jim Bergralis and Lin Xu, and Jeremy Simon from Dana-Farber. I also want to thank the support from AACR for this project. Thank you.
David?
I was just wondering if you actually just supply the downstream prostaglandin, is that sufficient to induce belzutifan resistance?
Yes, so that's actually exactly what we're doing. So far, data looks promising. So prostaglandin actually can induce also resistance as well. It's similar in fashion as PTGS overexpression, as you would expect. Yeah. So I didn't show the data, but actually we did also reverse weight upon the PTGS depletion. So we also saw the reverse phenotype as well. So.
How do these score in Jim's PDX models where HIF2 resistance mechanisms were seen before?
How did the
PTGS and the other molecules score?
Yeah, we haven't looked at that actually specifically. This is actually fresh new data. So I have to double check on the PTGS expression level in those resistant. But keep in mind, those actually PDX models are all intrinsic resistance. This PTGS actually is adaptive resistance. So it could be a little bit different. So I'm not sure whether that would score, but that's something we will look into. Yeah, that's a great question.
If I could ask the final target that you showed, the tissue distribution of INSRR?
Yeah, we actually don't know at this time because it's actually very little studied. It's not insulin kinase, but rather it's insulin-like receptor. So we know that at this time that is kinase activity could be important. We think it's the substrates that contribute to these phenotypes. The molecular mechanism, we don't know yet. But the phenotype is there, is very robust, with actually a couple of independent sgRNAs. We're actually pretty excited to see how that would work in terms of molecular mechanism. Yeah.
Great.
Thank you.
Okay, thank you very much. I will introduce the final speaker in this session, Jim Bergralis, who is at UT Southwestern, and he'll be continuing the theme of resistance mechanisms to RCC treatments.
Thank you. Thank you for the invitation and welcome everybody. I'm going to apologize to those of you who are on that side of the screen because I only have one pointer and that's a little farther away. So we're all very familiar about the importance of HIF, and I'm not going to belabor the point. you may know that the HIF-2 gene was discovered by Steve McKnight, among others, at UT Southwestern. The gene encodes a protein that binds HIF-1 and this binding is necessary in order for HIF-2 to bind DNA and induce transcription. Rick Bruick and Kevin Garner at UT Southwestern found a small cavity within the PAS-B domain of HIF-2 that led them to hypothesize that this could be targeted through small molecules. They used our library of compounds, they found the small molecules. This led to the founding of Peloton Therapeutics across the street from UT Southwestern, our biocenter. And Peloton Therapeutics developed this series of compounds. They are called PT because they are Peloton Therapeutic compounds. There is the PT2385, which is the first in human, the PT2399, which is the tool compound we use in animal models, and the PT2977, which is belzutifan, after the company was acquired by Merck. I want to point out that the numbers that you see here indicate the number of iterations of these compounds. So the company went through several thousand iterations of these compounds before they arrived to the clinical compound. So we went, as Ching alluded to, we went forward and we wanted to see whether this was going to work for kidney cancer. And Bill Kellen and I tried different models. In our case, we used a large platform of patient-derived xenografts or tumor grafts that we tested this HIF-2 inhibitors in. This is one example here, and I'm going to show you later the same example developing resistance. But you can see the growth of the tumor with vehicle, the effect of sunitinib, and then the effect of the PT2399 drug. And we did this at a large scale, almost 300 mice from 22 independently derived tumor graft lines, including 18 that were clear cell renal cell carcinoma. And overall, the PT2399 had greater activity than sunitinib and was also better tolerated, as determined by looking at mouse weight. Now, as you know, for targeted therapies, resistance is very common. So we asked, is resistance going to develop? So we took the same line that I showed you before, and we treated with the drug for a long time. And Ching already showed you some of this data. So this was a sensitive line, and you can see the tumor grew and then came down. But then after treatment for 6 months, the tumor got to be quite large. When we did immunoprecipitation analysis, we could show that prior to resistance, the inhibitor was able to dissociate HIF-2 from HIF-1 but following resistance, the complex had reformed. And we found in one of the lines a mutation in HIF-2 in the binding pocket, and in the other line, we found a mutation in HIF-1 in the surface between HIF-2 and HIF-1 And we believe that this mutation preserves binding despite drug treatment. So this set the foundation for the phase 1 clinical trial, which was run by Kevin Corney at UT Southwestern, who is here in the audience, with this first-in-class, the PT2385, which was subsequently improved and became belzutifan. But this already shows some proof of activity. We had a correlative study where we were asking patients if they would be willing to participate and give us samples. And we evaluated pre-treatment samples, on-treatment samples, and after the acquisition of resistance. And this is an example of what happens on treatment. So you can see 2 different patients. Each dot represents the HIF-2 complex. And as you can see, with treatment, there is a decrease in the number of HIF-2 complexes, which is what we would expect the drug would do. It dissociates the HIF-2 from HIF-1 and that leads to fewer complexes. And this also decreases gene expression. But we also asked some patients if they were willing to let us biopsy samples after the acquisition of resistance. This is an example of a patient of mine who was on treatment for about a year, who eventually developed resistance. We biopsied this tumor, and interestingly, we found the same mutation that we had found in the tumor graph in this patient. And this mutation led to preservation of the HIF-2 complex despite treatment with an inhibitor and reactivation of the HIF-2 gene expression program. And we found the same mutation in another patient that we also looked at. So the conclusions for this first part of the talk is that these PT drugs bind HIF-2 induce a conformational change, dissociate HIF-2 from HIF-1 and inhibit HIF-2 transcription. As for other targeted therapies, tumors develop resistance to these drugs. Resistance results from mutations in the binding pocket or in the surface between HIF-1 and HIF-2 And the development of resistance mutation shows that these drugs act by inhibiting HIF-2 and that HIF-2 is a core dependency. Finally, this shows that tumor graft models can predict resistant mechanisms in patients, which is going to be important for the second part of the talk. So here, we're going to be looking at resistance to Rapalox. I realize that these are drugs that we don't use very much anymore, at least in isolation, but it's an interesting question from a biological perspective. So there are two drugs that are FDA approved for kidney cancer, temsirolimus and everolimus. Importantly, temsirolimus becomes sirolimus or rapamycin. This makes up 70% of the circulating drug. This was approved initially in 2007, temsirolimus was. Today, most often used in combination with lenvatinib. Resistance uniformly arises, but it's been a mystery how it arises, and this is being looked at by the Harvard group, among others. So there's a paper from Sabina Signoretti, David Kiwatowski, and Tony Chwery, and they conclude this is looking at 6 patients that develop acquired resistance. They did not find mutations in mTOR, which is the target of rapamycin, right? In fact, mTOR is called mammalian target of rapamycin because rapamycin binds mTOR. So they look and they were surprised that they did not find a mutation in a single case. So they concluded that mechanisms of resistance are not easily explained by mutations, and they are likely dependent on more subtle transcriptional and other epigenetic changes. So the data that I'm going to show you is summarized in this slide. So we decided to use the same platform that we used to understand resistance to belzutifan, or the PT drugs to understand resistance to Rapalox. So we took mice, we treated with rapamycin, the tumors regress, but then if you treat for long enough, then the tumors grow. And then, interestingly enough, now if we took this tumor that was resistant and we put it into another mouse, the tumor became sensitive. And if we took these cells and we look at mTOR inhibition, in culture they were sensitive. So that was the first clue that this was not genetically a genetic mechanism, right? So I'm going to show you that how rapamycin is acting is by targeting primarily the tumor microenvironment. And the data that I'm going to provide, so what we did is we engineered the recipient mouse with a resistant mutation. So now if you treat this mouse that has been transplanted with the same tumor with rapamycin, you can still inhibit tumor cells, but you cannot inhibit the stroma. You cannot inhibit the tumor microenvironment because the mouse has a resistance mutation. And I'm going to show you that this is actually sufficient to confer resistance. I'm going to go fast through here because I know I'm going to be short on time. So this is the data, and I'm just going to show you a little bit of the data. This has been published earlier this year. So here we go. You have 3 different tumor graph lines. This is the vehicle curve in black. You treat with rapamycin, you can see that there is very substantial inhibition, but over time, in every case, with different latency, resistance develops. Interestingly, resistance develops despite persistent inhibition of mTOR in the tumor. There is persistent inhibition of the mTOR despite the fact that these tumors are growing. Okay, so I'm showing that here. So interestingly, however, while the tumor cells, which are labeled here in PAX8 with the PAX8 marker, don't reactivate mTOR when resistance develops, the stromal cells do. So resistance is associated with reactivation of mTOR in stromal cells, but not tumor cells. So that suggested to us that the cells of the microenvironment were playing a role. So what we decided to do is engineer a mouse with this mutation. I'm not going to go through the details, but this mutation interferes with drug binding. And when we engineer these mice, so we have now mice that have an mTOR resistance mutation that are being transplanted with the same tumor that you saw before. And you can see here, this is when you treat the wild-type mouse with vehicle, when you treat with rapamycin. And now if you do the same experiment in the mutant mouse with either 1 allele or 2 alleles that are mutant in the green or purple, you can see that in every instance we're inducing resistance. So inhibition of mTOR in the tumor microenvironment is necessary for the activity of Rapalox. And that's probably why we don't see mutations in patients. So I'm going to summarize here. So unlike for the HIF2 inhibitors, resistance to Rapalox is not caused by mutations in the target. So we are not claiming that mutations in the stroma are going to occur and are going to give resistance in humans. What we are claiming is that the activity of rapalogs requires inhibition of mTOR in the tumor microenvironment. And for reasons that are yet unknown, mTOR becomes reactivated in tumor microenvironment, and that appears to be sufficient to confer resistance. So I've shown you before with HIF2 alpha that these preclinical models are credentialed. We were able to predict the HIF2 resistance mutations, and we think based on what we've seen clinically that something similar may be happening in patients. I've emphasized that mTOR gets reactivated in tumor microenvironment, and that forced mTOR activation in the mouse is sufficient to markedly dampen the effect of rapalogs on tumor cells. And I'm just concluding, I want to make sure that this is the experiment, but I'm not claiming that this is the mechanism of resistance, because I think mutations in stromal cells are unlikely to be a mechanism of resistance because there is no proliferative stimulus. Boy, I'm surprised I'm still within the time range, I think. All right, so I'm just going to conclude. There are a lot of people that I want to thank for this work. So Ramesh Bhutti, along with Juan Yang here, were involved in the rapalog resistance project. Wenfang Cheng was involved in the HIF2 alpha resistant project. A number of people here maintain our tumor graph platform, and Roy published recently on this. I already acknowledged Kevin, who led the phase 1 clinical trial. We work very closely with Payal. And you've seen also, we work closely with other members. You've heard from Ching, and you'll hear from several other people from UT Southwestern, of course. We work with Peloton, and yeah, I want to thank especially patients and their families. Thank you.
Okay, Jim, if it's okay, I'll start the questions. Sure. So, you know, obviously there have been patients that have been treated with rapalogs over the years, and perhaps banked samples that could be queried in such a way, you know, now with spatial approaches, laser capture, et cetera, the stroma could be investigated for acquiring these kinds of changes. Is that feasible?
Thank you. So in fact, we had so the challenge there is that you would need samples of patients that have been treated, that are currently on rapamycin, right? Because if there is no rapamycin on board, seeing mTOR reactivation is what you would expect. So we had such a trial. We had a trial where we were giving patients everolimus, and then we were collecting samples while the patients were on drug at 2 different time points. Fortunately, that trial didn't accrue a lot of patients. We only have a handful of patients. The data is consistent, but it's not a trivial question to address. What I can say, and that was the paper that the Farber group published, is that they had 6 patients that had a beautiful response. They acquired resistance. In none of the patients, they found an mTOR mutation, suggesting that the mechanism is not mutation-driven. And of course, for us, the fact that you lose resistance when you transplant the tumor, you lose resistance in vitro, is also consistent. And we've done mutation analysis, and we don't find mTOR mutation. So I think, you know, I would say, you know, it's unlikely to be a genetic mechanism. It would be great if we could validate this in patients. May not be trivial.
Yeah, I understand that. Other questions?
Yeah, thanks for your great presentation. So you have a hypothesis of what cell types in the microenvironment are being acquiring resistance?
Right. So I have a postdoc in the laboratory who loves calves, so he thinks it's the fibroblasts. We don't know yet. I mean, there are a lot of questions that I mean, we don't really have a clue about the mechanism, for example. I think the power of this is that the experiment is a genetic experiment, right? So the only thing we did is we mutated 1 amino acid in the NOD SCID mouse strain. And then we transplanted the same tumor, and we got resistance. So I think the data unequivocally shows that the activity of rapalogs requires mTOR complex 1 inhibition in non-tumor cells. It is a speculation that is the cells of the tumor microenvironment, but I mean, we targeted all of the cells in the mouse.
Jim, really interesting findings. So I'm thinking in terms of the mechanistic aspect over here, right? If the mTOR is still shut off in the tumor cell, the nutrient sensing is still absent in those cells. What are the fibroblasts feeding to the tumor to keep it alive?
Exactly. So great question. I mean, that's also something that is quite puzzling. It was very puzzling for us. The fact that, I mean, as you saw with HIF2, what happens is you mutate HIF2 and you recover HIF2 function, right, in the tumor cells, but you are not seeing this here. mTOR complex 1 remains off in the tumor, right? So as you correctly point out, the reactivation of mTOR in the stroma is sufficient to compensate for the loss of mTOR complex 1 function in the tumor. How that happens, we don't know. But I can tell you that if we do co-culture experiments, so we take tumor cells and we culture them with fibroblasts, getting back to the prior question, that have this mTOR mutation and we treat with rapamycin, in that co-culture experiment, the signal is not as robust as what we see in the mouse, but it appears that the presence of the resistance fibroblasts can confer some resistance to the tumor cells. It's the same principle. So we would argue that the effect is in trans or paracrine, yeah.
Jim, if I understood correctly, the initial experiments were done in immunodeficient mouse. Do you think the similar mechanisms would be elicited if you were in an immunocompetent background? And do you have any thoughts about how the tumor microenvironment, the immune microenvironment might be contributing?
Right. Yeah, all of the experiments for both models involve PDXs, so the mice are uniformly immunocompromised. I mean, obviously we've generated genetic models as well of kidney cancer, but we are not using these in this context because we think that this is the more physiological context to understand resistance in humans. So I cannot exclude the potential role of the tumor microenvironment. I think what the data argues for is that simply substituting one amino acid in the mTOR gene that renders the mTOR protein unable to bind rapamycin is sufficient in one instance to completely abrogate the effect of rapalogs on tumor growth, and in two other instances to substantially dampen it. So that data suggests that mTOR inhibition in the tumor microenvironment may even be more important than mTOR inhibition in the tumor itself. Because the model is immunocompromised, I cannot say anything about the immune system, but if you ask my own opinion, I don't think it's going to be it's going to play a major factor. I think this is just showing, in an immunocompromised host, just mutating mTOR is sufficient.
I had a question. Have you looked, or do you think that the same mechanism comes into play for patients with TSC or AMLs where we treat with monotherapy, everolimus, and 2? Have you considered using those to sort of validate some of these mechanisms biologically?
Right. So I think, that could be a different context because you have, in that context, the mutation is germline. And I think the dependency of mTOR may be greater. So in kidney cancer, is not clear whether, I think it stands to reason, but I wouldn't say that the data supports this, including data from some of you in the audience, whether mutations in mTOR complex 1 regulators, such as TSC1, TSC2, or REB, would necessarily increase sensitivity to mTOR complex 1 inhibitors. So I think, if I were to speculate, I might expect that if one were, in fact, in thyroid cancer, there is at least one publication showing mTOR mutations associated with resistance. Assuming that that's generalizable, I would say in familial syndromes where you have mutations in TSC1, I would hypothesize that that might be the more likely mechanism compared to what we see in sporadic kidney cancer. Thank you. And I mean, I think that in VHL patients, resistance to the HIF2 inhibitor is observed, but I think it takes a lot longer than what we might see in the sporadic setting, or that what we see in the sporadic setting. So I think, the germline and the sporadic setting are very different settings.
Okay, I think we have to conclude. We'll have a 15-minute break, and then the next session will take place. And let's thank Jim and all the speakers for a very stimulating session.